Today we are going to see another important concept, that is chapter 5, Principles of Inheritance and Variation. Actually, it is about genetics. You might have studied about what is genetics in your lower classes. Actually, genetics means the branch of biology that deals with the study of heredity and variations. So, in short, we can say genetics is equal to heredity plus variation. So, what is heredity? That is the traits, the specific traits which are transferring from parents to the offspring or one generation to the next generation. Now, we will see that what is the principles on which basis the inheritance and variation is happening in the living organisms. What is inheritance? In your book, page number 69, it is given clearly, that is the third paragraph, fifth sentence. Inheritance is the process by which characters are passed on from parent to progeny. Actually, it is given one important key concept, that is inheritance is the basis of heredity. Some information, some genetic information is transferred from parent to offspring or from one generation to the next generation. Here, what is variation then? Inheritance, transfer of the traits from parents to the offspring. And what is meant by variation? Here, the degree by which the progeny differ from their parents. How much percentage differences the progeny or the offspring or the next generation is showing, just when compared with the parent. That is called variation. Why we need to give importance to the variation? Because variation, it leads to the evolution. Variation leads to the evolution. So here now we'll see that who is the hero of this chapter. Here he is none other than Gregor Johann Mendel, who is the father of genetics. I think in your lower classes you studied who is Gregor Johann Mendel and what is his experiment and what was his findings. Mendel and his work on heredity. And here the first quantitative study of the inheritance was carried out by Gregor Johann Mendel. And he chose garden pea plant. That is the edible one, garden pea plant, because he selected 7 distinct traits from 34 varieties of garden peas. See, just imagine 7 distinct traits what you studied. That is the trait is seed color, seed shape, and then flower color, and then pod color, pod shape. That is the shape. Green or yellow or inflated or constricted, and then flower position, whether it is axial or terminal, and then plant height. That we are very familiar with the plant height, the term tall and dwarf, which is in the monohybrid, just a test cross, we know very well. And here why he chose pea plant, that is Pisum sativa, because of the reason. What are the following reasons? You studied already in your lower classes, but just a recall. Why he chose Pisum sativa, that is pea plant, garden pea plant? Because first, point number 1, there were number of varieties available with the distinct characteristics or traits. So for example, green seeds or yellow seeds. So first point, distinct characteristics or number of traits, variety of different types of characters are observed in the plant, so that it is easy for the researchers or one who is conducting the experiment to. So here Mendel preferred this plant for that reason. Second one, the flower of the pea plant is so structured, that is the stamens and pistils are completely enclosed by petals. What is the benefit of this? What is the benefit? Can you imagine? Because self-pollinated. Very good. Because no other go. Okay, no other go. Can you just recall what you studied in the chapter 2? You studied about clistogamous and this chasmogamous. Here, like that, in that category only. Okay, clistogamous. Here, the stamen as well as the pistils are completely enclosed. So, no other go, the flower has to undergo self-pollination. So, anthers burst within the bud. So, the stigma is covered with the pollens even before the flowers open. Then, the third point, pea flowers could be easily cross-pollinated. Okay, because now when we do the artificial hybridization, we need to look into two important concepts, that is one is emasculation, another one is bagging. So emasculation means removal of anther from the flower, and bagging means that's after dusting the pollen on the surface of the stigma, we have to tie with the bag. Okay, so artificial hybridization is easy in pea flower. So it was done by removing the stamen, and then that is what I say. Then fourth point, it was easy to culture because it is an annual plant, so it can give the yield quickly. The plants in the open field and in pots also, because not only in the field, in the pots also it is easy to grow. Then here next what Mendel observed in the plants as a plus point. When hybrids were mutually crossed, they resulted in perfectly fertile progeny. Because to just start the experiment, Mendel cultured many pure line breeding. Okay, pure plants. Pure breeding means what? Because the plants for many generations, they were keeping on undergoing the self-pollination, so that 15th generation or 16th generation, or when when we got the desired character, we'll be able to say this is the pure line or true breeding. Okay, pure line or true breeding. So here, just in order to get the pure lines, the plant should be fertile. It means that it will be able to produce the seeds for the next generation. So here the fertility is possible. So that's why it was selected. Then the pea plant relatively had short period of growth. As I already said, it can grow well within a short span of time, so that we can make use of this plant for the best experimental research. Here, for these reasons, Gregor Johann Mendel selected this plant. Then I think you know very well. Then just it is given Mendel's laws of inheritance, which is given in your book, page number 70 under 5.1. What you studied in your lower classes, please repeatedly I have to say, please you recall, because you studied about this in 10th standard. What is the experiment? In what way it differs from other biological experiments? Here biology, it meant only for the living. Okay. Here Mendel's laws of inheritance. Here all the experiments done by Gregor Johann Mendel, it was a carefully planned one. Then he studied only one trait at a time. He didn't confuse by just mixing with the more traits. Of course, he discovered 14 contrasting traits, but no, he just experimented with only just one trait at a time. And he studied specific traits and performed experiments with utmost care to prevent contamination. What would happen if there is the flower is contaminated? Because the result will not be an accurate one. So we'll get some different traits. So that it is just apart from the true breeding, that's a characteristic feature. And he kept accurate records and analyzed them statistically. Here you have to understand that in what way, and this experiment is different from that other biological experiment. Under 5.1, that paragraph, here it is given, it was the first time, I think first, second, third, fourth, fifth, sixth, and the 8th, 9th line, or 8th and 9th line. During Mendel's investigations into inheritance patterns, please you underline, it was for the first time that statistical analysis and mathematical logic were applied to problems in biology. In this way, this Mendel's experiment is entirely different. Is that clear? Statistical analysis and mathematical logic were applied to the problems in biology, because his experiments had a large sampling size which gave greater credibility to the data. Because with the 7 different contrasting pairs of traits, he was able to collect much more information, so that he just implemented with the mathematical logic